Hello guys, welcome to IB360. Today we will start with protein versus polypeptide. In my last class I had described amino acids in details. So today we will differentiate between these two terms which are often interchangeably used, polypeptides and proteins, and in the meantime we will know about them individually. So before going into the difference between proteins and polypeptides, let us understand how peptides are formed. So peptides forms by condensation of amino acids. Peptides form due to condensation of amino acids. So two such amino acids or more will undergo condensation and eliminate a water molecule in the process, and a peptide linkage will be established between the carbon of the carboxyl group and the nitrogen of the amino group. Okay, so here usually there is an OH present, and that is how H2O gets eliminated, and this is the peptide linkage which is established. Another important thing which you must have noticed is N terminus and C terminus. In every primary protein structure rather, that is, there are various organizations of protein, primary, secondary, tertiary. You will see these things later on. So in primary structure, when we talk about primary protein structure, we usually have an N end and we have a C end. That means this is the terminal amino acid on the left side and this is the terminal amino acid on the right side. This is the convention, and that is usually because the left side amino acid starts with the amino group and the right side amino acid, the right terminal amino acid, ends with the carboxyl group. So that is why N and C, left side N and right side C. This is the end actually. These are the terminal amino acids, these two, the terminal amino acids. And we know that polypeptides or proteins, whatever they are, they would be chains of amino acids. They would be heteropolymers of amino acid. Both polypeptides and proteins are heteropolymers of amino acids. Why heteropolymers? Because the various amino acids, they wouldn't be same. Some would be serine, some would be leucine, some would be phenylalanine, some would be lysine, and this will go on. It's not that all are glucose or something like that. So the monomers, they wouldn't be identical. So the polymer wouldn't be homopolymer, rather a hetero prefix would be used. As simple as that. So this is how a peptide bond is formed. This bond is basically amide. Okay, peptide bond is an amide bond. Okay, and this is the basic bond that must be established between amino acids to form proteins or polypeptides, whatever. And the bond is the NHCO bond. Okay, so if the bond is formed between two amino acids, it would be a dipeptide. So this is the first point. This is the second point, and this is the third point. The fourth point is, if two amino acids, then a dipeptide. If three amino acids, then it will be called a tripeptide, the entire unit. So like this, it will go on. If more than three, a few, then oligopeptide, and numerous, polypeptide. Few amino acid, oligopeptide, and more polypeptide. And occasionally the peptide is acidic or basic due to presence of specific amino acids, but that we will talk about during classification of polypeptides. So this is the concept of polypeptides. Okay, there would be an amide bond or a peptide bond between the individual amino acids, and that will involve elimination of water molecules, so the process will be a condensation process. They will definitely have an N end and a C end, and it will be a and polypeptides are hydropolymers of amino acids. Then what is the difference between polypeptide and protein? So you see, polypeptides are when many amino acids, they are establishing peptide bonds or amide bonds between themselves by eliminating hydrogen atom between the carboxyl carbon and the amino nitrogen. Then that is polypeptide. But proteins are basically the larger versions of polypeptides. Proteins are made by one or more polypeptide chains, and hence they will have a higher molecular weight than polypeptides. Also, since proteins show various degree of folding, a primary degree or a secondary degree or a tertiary or a quaternary, so it shows a various or varying degree of folding, intra-protein foldings basically. So they will involve other bond formations also, other than the condensation, other than this particular bond, peptide linkage. There would be other kinds of bonds like a hydrogen bond or a disulfide bonds, other electrostatic interactions, and these all bonds can be intramolecular or in fact sometimes intermolecular also. Intermolecular means between two different or separate protein molecules, and intramolecular means within the same protein macromolecule. And these various bonds results into varying degree of folding, and they govern the three-dimensional structure in contrast to polypeptides. Polypeptides are not three-dimensional because they are not showing the folding process which we see in proteins. Okay, so proteins are large-sized heteropolymers. Large size means macromolecules, and hetero because the amino acids which are its monomers are obviously not identical, and poly because many amino acids, in fact amino acids will form peptides, and many peptides which are forming the polypeptides, they will form the proteins. Okay, so they will be formed with one or more polypeptide chains. In fact, a single polypeptide must be at least 50 amino acid long in order to qualify for the term. Okay, at least 50 amino acids are must for a polypeptide to be called so. At least 50. And on the other hand, if we talk about proteins, they will have thousands and thousands of amino acids. But polypeptides, they must have at least 50, a single polypeptide. Okay, a single polypeptide must have at least 50 amino acids to be called so. Now, a protein which has a single polypeptide is called a monomeric protein. And monomeric proteins examples would be myoglobin or ribonuclease. Ribonuclease is an enzyme which breaks down RNAs. Okay, so this is one point. Second point is the single polypeptide proteins. One polypeptide protein is a monomeric protein. Example is myoglobin is basically the hemoglobin for muscles, and also RNase. RNase is ribonuclease. Okay. Then if two polypeptide proteins, then dimeric protein. A very famous example, insulin. Now insulin, insulin is a very famous enzyme. It is secreted by the beta cells of pancreas. Okay, so 4 polypeptide proteins are also there, and 4 polypeptide protein is a multimeric protein, and its example is hemoglobin, the oxygen carrying protein present in RBCs. Hemoglobin found on surface of RBCs, we know they are the oxygen carriers, the transporters of oxygen in the form of HbO2 from lungs to tissues which require them. So these examples you can try to remember. Okay, so this is the concept of multimeric proteins. Multimeric means there would be more than one polypeptide chains involved in it. Now comes to very famous examples, collagen and rubisco. So collagen is the most abundant protein in animal world. Okay, collagen is the most abundant protein of animal world, while rubisco, which is ribulose bisphosphate carboxylase oxygenase, you will soon come across this term rubisco when we study photosynthesis in plants. Very important, significant role it plays there. So rubisco is not only the most abundant protein in plants but also the whole biosphere. Collagen, most abundant in animal world. Rubisco, obviously most abundant in plants, but also the whole biosphere. So if someone asks you what is the most abundant protein, the answer would be rubisco. If they are more specific, they say plants or animals, then accordingly your answer will change. So rubisco is a very important example of protein. And this is all about protein versus polypeptides. I hope you understand.